Tuesday. So we just finished talking about cortical magnification. Need to finish up a couple of bits. We didn't do this part yet, right? Genes in the environment? No, I didn't think so. All right, so one of the central questions in psychology often has to do with is a behavior the result of our biology, or is it the result of our environment, so our upbringing, that sort of thing, or because we were raised in a period of lean times or good times or with siblings. There's all sorts of questions about how our environment might affect our behavior. This is a pretty central question we'll talk about throughout the semester. It turns out, of course, that nothing is that simple, that these are not as independent as one might believe. That is, your environment actually shapes your genes in ways that we really are just starting to understand, and it's a field we call epigenetics, and we'll talk more about that here in just a moment. So basic review for pretty much all of you, I would hope, that you all know what a gene is. It's the basic unit of heredity, hereditary transmission. These are sections on strands of DNA, which is, of course, organized into chromosomes. We have 23 chromosomes, 23 pairs of chromosomes, I should say, which comprise, which make up all of our genetic structure. Now, one of the things we're starting to understand is that those genes can be turned on and off. They can be altered slightly through our experience. Chromosomes are, of course, these strands of DNA wound around each other in a double helix configuration, which Watson and Crick, of course, very famously won the Nobel Prize for, despite the fact that they weren't really the first people to come up with the idea. As we move through the semester, again, this is mostly laying groundwork for later work we will talk about. We, as psychologists, will often try to come up with ways to study genetics in a way that doesn't require us all to be molecular biologists. So, and in fact, a lot of the work on sort of genetic heredity predates any of our knowledge of the human genome. So most of what we talk about has to do with looking at degrees of relatedness. So we look at one of the simplest ways is monozygotic versus dizygotic twins. So monozygotic twins are identical twins. They share the same DNA, versus dizygotic twins who are basically no more related than just a basic brother and sister. The assumption, though, is that monozygotic and dizygotic twins share the same amount of environment, right? So the monozygotic twins share genes and environment. Dizygotic twins share some genes, but all the same environment. And so if monozygotic twins show more degree of relatedness in a trait, so something like schizophrenia, we can probably conclude that there is a genetic component to that trait. Whereas dizygotic twins, in that case, have the same level of risk as siblings do. So that reduces the probability that we're talking about some sort of environmental influence. So this allows us to look at genes and environment in different ways. So we talk about heritability. So we measure, which is a measure of the variability of behavioral traits among individuals that can be accounted for by genetic factors, details of which we don't need to get into, but you can essentially establish a probability component. That is, there is sort of a 40% heritability, that sort of thing. So we'll spend some time looking at things like twin studies in a couple of areas. We'll look at intelligence, we'll look at personality, and we'll look at sexual orientation and how it relates in terms of the level of heritability. But that's usually the way this is done, is by looking at people with relative amounts of heritability. So you can look at monozygotic twins versus dizygotic twins. You can look at siblings. You can look at adopted siblings, which of course have no genetic relationship. And that allows you to sort of make estimates about heritability as well as environment. The final note I want to make here is an area which I have been trying to accumulate more knowledge in, but it is very new. This postdates my graduate training for sure. And there are certainly better experts in the department that I am in this area, but there is a lot of really interesting work being conducted in epigenetics. And this is the emerging field of study for examining how our experiences shape our genetic expression. So the thing your genes do for us is they act through protein expression. So you create proteins, and this is basically how all of our genes are making everything about us tick. One of the things that happens is our experiences can alter that expression. And what we're now learning is that those alterations can then be inherited. That is, if, give you a fairly simple example, your grandparents used heroin, that will have altered their genetics, that your parents would have, one of your parents would have inherited, that then you would have inherited. Stress, high levels of stress, people in war zones, people in poverty. That level of stress changes your genetic expression, and that is inherited from one generation to the next. And so we'll start looking at some of these areas, and it's very new, very strange in the way that this is all happening. So, for example, in my physiological psychology class, we read an article where they looked at rats or mice, I can't remember, some sort of rodent. They're usually used as models for humans because, frankly, they're cheaper and you can do things to them you can't do to humans. And what they discovered is that the male offspring of rats that had been given sort of copious amounts of cocaine, the male offspring of those rats had epigenetic alterations, and those rats were much less likely to prefer cocaine. So it was a sort of ironic epigenetic alteration by which it downgraded the response of those rats to cocaine because their fathers had actually been exposed to quite a bit of it. So this is an area where we have a lot of work going on, really interesting things in the field that we'll start to learn more and more about. I'm actually going Wednesday afternoon to a talk at the the Association for Psychological Science on stress, poverty, and brain development, and there is some epigenetics in that area as well. Questions about that? That's going to come up again in a couple of areas. Well, to finish up our basic introduction to neuroscience, I want to talk a little bit about how we investigate the brain. We're going to kind of skim lightly through this discussion. But much research in neuroscience correlates loss of specific functions to brain damage. This is actually an area we call neuropsychology. So to give you a sort of taste of how this kind of research has been done over the centuries, Wernicke and Broca were the first two people to engage in this kind of research. Wernicke discovered a part of the brain responsible for language comprehension, and Broca discovered another for language production. And in fact, when we get to talking about language, we'll talk about Wernicke's aphasia and Broca's aphasia. So people with Wernicke's aphasia cannot actually comprehend spoken language. People with Broca's aphasia can understand language but can't actually produce it back. And so it shows there's sort of dissociable areas of the brain involved in different aspects of language. Phineas Gage will actually delve into looking at Phineas Gage in more detail when we get to talking about emotions and how the frontal lobes are involved in emotional control. Essentially what happened with Phineas Gage is he had a terrible accident in which a railroad tamping, he was a railroad worker, and they were actually using a rod to tamp down explosives into rock. The explosives went off, the tamping rod went, well, let me go forward here to the next slide. This is Phineas Gage. This is actually a reconstruction by Hannah Antonio Damasio in a book called Descartes' Error. But essentially this tamping rod you have in his hand, or he has in his hand, went through his orbital socket, through his left eye, and out the top of his brain. He lived, which is pretty remarkable considering this is the late 19th century, but it dramatically altered his personality. And so what we saw with Phineas Gage is that by disconnecting the frontal lobe from the amygdala, he was actually no longer able to control his emotions and completely lost all of his rationality. Other work has to do with the lateralization of the two hemispheres. When we talk about brain lateralization, we mean that one task tends to be more often in one side than the other. So a lateralized function would be something like language. For most of us who are right-handed, your language tends to be in your left hemisphere, almost entirely your left hemisphere. Those of you who are left-handed, you might actually have less lateralization. In fact, most left-handed people have language in both hemispheres, which is a bit of an advantage. So if you have a stroke, you're probably less likely to lose your language abilities. It also means that most neuroscientists aren't interested in you being in their studies because we insist on everyone being right-handed because left-handed people have such different brains from the rest of us that we just like to ignore that they exist. So it's sort of an interesting part of neuroscience is it's almost entirely about right-handed people, which isn't fair, but it's kind of how it works. One of the other ways this has been studied, though, is by using what's called a split brain procedure. We've gotten past doing this as much at this point. This is something that was done before in the past to try to treat people with epilepsy. So they would actually sever what's called the corpus callosum, which is the part that connects the two halves of the brain. And so they would sever the connections between the two hemispheres, which meant they could no longer communicate with one another. As a result of these studies, we find, again, for most people, the left hemisphere tends to be more verbal. This is where most of our written and spoken language occurs. It's generally where all of our linguistics happens. And the right tends to be more visual spatial, certainly far more spatial. So putting together that IKEA furniture does not generally involve the left hemisphere because there are, of course, no written instructions. It's all spatial, right? It's all visual spatial. There is some evidence that this is one of the reasons why we see some gender differences in cognition. So, for example, women tend to have, in general, larger left hemispheres than right. Men tend to have larger right hemispheres than left, which is why men tend to be more often left-handed. It also seems to be why women tend to have better verbal abilities and men tend to have better spatial abilities. Again, looking at population means. So if we look at a split brain patient, this is one of the ways in which these people are studied. You can see they have no connection between the two hemispheres. I like they added a ponytail. That's a nice detail. So they're asked to look here at this fixation point. Now, remember, everything that's over here in the right visual field is going to the left hemisphere. So they'll be able to say that they see a ring. They won't be able to say anything about the fact that they see a key, but when they're asked to pick up the object shown with their left hand, they pick up the key because that's over here in the right hemisphere. This information has gone over the right hemisphere, and as a result, that's what they can find with their left hand because that information about the key is only over there in the right hemisphere. So that's some of the ways in which we've been able to look at patients with brain damage. Trying to study links between brain structures and behavior in individuals without brain damage is also an area we look at. So this is where we record electrical activity in the brain, primarily using a technique called electroencephalography. Electroencephalography is where you just basically take electrodes, put them on somebody's scalp, and you can actually record the electrical activity in their brain. It's a little more complicated than that, but it's really pretty much that simple. In some instances, in animals in particular, you can actually record the activity of individual neurons. It's done in humans very much because you have to have a reason to cut open their skulls. They don't let us do that to just anybody. So sometimes this is done in patients who are undergoing neurosurgery already, but primarily it's done in animals. We'll talk more about that later. But electroencephalography is a way in which electrical activity in the brain is studied by a lot of people because it's an easy technique to employ and it's also really cheap. Top-of-the-line EEG equipment, which is the same used in what we call event-related potentials, it's going to run you maybe $100,000, and you can run participants for as long as you'd like. By contrast, the other way to study electrical activity in the brain is usually using a technique called magnetoencephalography. That price tag is in the tens of millions. You have to build a structure around the thing. It's a little crazy. Not a lot of people doing that work. But lots of people doing EEG work. It's a really great way to get involved in neuroscience. My dissertation, I actually did this event-related potentials, where you time lock these electrical recordings to external events, and you can actually see differences in how people respond to different stimuli, and you can actually see that in real time. So the great advantage to This particular technique is you get millisecond by millisecond precision that you can't really get in any other technique. So this provides us with some pretty good information. In fact, in my seminar, we just read this really interesting paper about where they looked at perception of weapons in officer-involved shootings, and they were actually able to show this switching of attention related to weapons effects and how it happens automatically. So this is what the data looks like. Put all these electrodes on somebody, and then you can see this is the live recording. This is where the participant blinked or moved, but this is sort of their basic recording here. Anyone done a sleep study? Can I do that? Yeah, they hook you up something like this. Probably not that many electrodes, though, right? No, probably just a few. Yeah, this is usually done. The denser the electrode array, the more information you get. So if you're interested in cognition, they'll usually do a lot more electrodes. When we get to talking about sleep, we'll very briefly talk about these brainwave patterns that are associated with sleep, because they change as we move from being awake to being asleep. So some other neuroimaging techniques, briefly talk about. We're using more advanced technology to create images, again, of the living, healthy brain. EEG is great because we can see the brain more in action, but these techniques allow us to actually see inside the brain into specific structures. So some structural techniques allow us just simply to look and map the size and connectivity of brain structures. So there's been some work, for example, on poverty, stress, and brain size and intelligence, and so they can just use these structural techniques to do that. The advantage of these structural techniques is they're very precise, and they're much easier to accomplish and a little bit less controversial because you're looking at brain structures. You're not trying to correlate activity, but merely look at the structures themselves. Functional brain activity shows activity while someone engages in a cognitive or motor task or any other kind of task. So the structural brain images we talk about are CT scans, which is simply a series of x-rays which are then used to construct an image of the person's brain, a 3D image of the brain. Less favored really anymore, well, they're cheaper and a little bit faster, but we're starting to learn that a full brain CT scan actually gives somebody a pretty high radiation dose, so a lot of people are moving more to MRI. MRI is, of course, magnetic resonance imaging. The great advantage of MRI is it actually images water, so it's actually able to look at soft tissue, which is why if you tear your rotator cuff or tear your ACL in your knee, they're going to send you to get an MRI because they can actually read that soft tissue. Well, the brain is entirely composed of soft tissue, so you can get nice structural images using an MRI. Now we can use the same equipment to do what's called functional magnetic resonance imaging. Functional magnetic resonance imaging, same basic idea, but instead of imaging where water is being used in the brain, or sorry, where water is in the brain, I should say, it's actually designed to see where you have been using oxygen. So deoxygenated hemoglobin, or deoxyhemoglobin, has a different magnetic property than oxygenated hemoglobin. They actually spin differently, so in the presence of a magnetic field, it changes the way they respond. And so we can actually look at the ratio of deoxygenated hemoglobin to oxygenated hemoglobin. And where you have more deoxyhemoglobin, the assumption is that's where your brain has been busy doing something. And so the assumption then is that where there's oxygen being used is where the brain's busy doing something. So that's the whole idea behind functional magnetic resonance imaging. Great thing about fMRI is you get pretty good pictures. You get pretty good idea about how it's unfolding over time. Not great. It's not super expensive. It's $300-400 a participant, so it's not cheap, but certainly less expensive than PET scans. And also, you can scan people as often as you'd like, so participants can come back again and again. PET scans are a little bit different. PET scans are positron emission tomography. Sometimes it's also called SPECT, which is single photon emission computed tomography. Basically, what happens with PET scans and SPECT scans is you are injected with a radioactive isotope, that will attach to different types of receptors in the brain or to different things in your blood. So PET scans can be used to figure out where your brain's using sugar, which is your brain uses a lot of sugar as it's processing and it's doing its stuff. PET scans can also look at hemoglobin. It can also look at where different types of neurotransmitter receptors are, so you can actually track where serotonin's being used in the brain, that sort of thing. Problem with PET scans is they're expensive. We're talking $4,000 or $5,000 a participant. The reason for that is you have to have a physicist who creates your isotopes, and then you have to inject the patient, and then it takes a lot longer to scan. They're also they're not as good in terms of the kinds of studies you can do, because you have to have participants doing the same thing for a long period of time. So they're not used as much in neural imaging. The number one use for PET scans now is they've come up with some very good radioisotopes that essentially attach only to cancerous cells. And so they do these whole body image scans for people who have had cancer to determine if cancers come back, because those radioisotopes will attach to those fast-growing cancer cells. So that's much more often the case for PET scans. Give you an idea what these images look like. It's basic CT scan. These are fMRI pictures. The most important thing to understand about fMRI studies is this brain you're seeing isn't anybody's actual brain. It's about 1,000 brains that have been averaged together. So this is sort of an idealized brain. So whenever you see this, this isn't actually happening in someone's brain. They've taken the data and put it on somebody else, on this model brain. But what they do is they show differences between different kinds of tasks involved in these scans. So the last thing I want to talk a little bit about is, so we've studied brain damage for centuries. We now have some new techniques for actually mimicking brain damage. In some of my studies, we've done it with drugs. So we've actually been able to induce temporary amnesia in patients using a drug called midazolam. But more often, this is done with what's called transcranial magnetic stimulation, in which they can temporarily deactivate neurons in the cerebral cortex. And so that then allows them to conduct experiments and find more causal explanations, because fMRI on its own is actually just a correlation, right? Because we've correlated brain activity with some sort of external activity. So what I thought we'd do is I would show you what transcranial magnetic stimulation looks like, so you can see for that. Also show you why you should be careful about where you go to grad school. This poor grad student's about to get his brain shut off. Temporarily, he's fine. RTMS is another technique. There's actually two sort of related techniques, RTMS and RTDCS, which is transcranial direct cortical stimulation, are being investigated as therapeutic techniques. So there are some evidence that transcranial magnetic stimulation is a potential treatment for depression. Dr. Green has actually shown that transcranial direct cortical stimulation potentially can improve things like creativity. But TMS in this instance is basically used to show what happens when you shut down part of the brain. So I don't remember which one's which here. So they've completely shut down what's called Broca's area, which is his ability to produce speech. They'll do it again here in a more compelling way. But again, this is why you have to be careful about where you go to grad school, because you'll end up being this guy.
Okay. And when I say stop, pull it out. Count. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30. Other side.
So casual about it.
Position. Count. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26. Stop.
So what you can see is they're able to show that disruption of Broca's area only occurs in the left hemisphere and not the right hemisphere. Why are we still doing that? Okay. So that's what transcranial magnetic stimulation looks like, where you can actually, and they've got very precise abilities to do this, so they can black out part of your visual hemisphere. They're able to briefly disrupt brain functioning, and they say it's perfectly safe, and the damage it's temporary, but